So today I'll be giving the next lecture in the Bio 410, Bio 510 Bioinformatics class at California State University in Monterey Bay. We're going to be talking about a couple of core concepts and fundamentals of how to use set theory to understand probability. And then we're going to be exploring different sort of perspectives on probability and how it's applied, specifically the frequentist versus Bayesian perspective. And we'll be detailing out exactly what a Bayesian approach is as well. As Bayesian approaches are becoming increasingly more important, especially in the life sciences, and will pop up later in this class. So let's just talk about some of the basics of probability. We're going to go over some specific examples. I'll give you some. We'll do some calculations and show you some numerical examples in class. But just to give some of the background information, right, when we talk about probability, like why is it important to us as biologists, as bioinformaticians? So as bioinformaticians, it's especially important to us because almost all of the tools that we use rely on some sort of underlying probability structure. Whether we're talking about e-values in BLAST or hidden Markov models for multiple sequence alignments or calculations of evolutionary distance in like a Jukes-Kantor model, right? We're always thinking about these probabilities. And in this case, like if we're thinking about DNA, we're always thinking about basically how, we've got this four-sided die, and every time we roll that die, we could end up with one of, four different bases, right? And so along those lines, right, so one out of every fourth rows, I would expect to be a T in this case, right? One out of every four would be a C or an A or a G. Those are the kinds of things that we're talking about. Like what's the probability, the chance that I'm going to see something? If we think about like a common case where we use probability every day, like think about your weather forecast. So you're going to look at some sort of weather forecast and it'll say, there's a chance of 40% rain. And so what does that actually mean? Like, what does 40% mean? Will it rain 40% of the time? Will it rain over 40% of the area? What is that actual definition? Well, it's actually based off of something called the probability of precipitation, or POP, and this describes the chance of precipitation occurring at any point you selected in that area. So if we think about a specific area, we can say, well, what's the probability that we would expect to see precipitation? And it's basically calculated using two numbers. One, the confidence that precipitation will occur somewhere in the forecasted area, so somewhere in the spatial area. So how confident are you there? And the percentage of the area that will receive measurable precipitation. So it's a factor of two of these things that is where we actually get this 40% number, right? So that's what we talk about when we talk about that percent of the area. It's actually a probability estimate. So something you see it every day, right? So how does what are the basics of probability? When we think about probability, an effective way to conceptualize it is to think of the basics of set theory. And so if we have a space S, and so we can consider that each one of these little boxes is a space S, right? And in each S, these are the set of all possible outcomes. And so within space S, we can have events, and those are sets of some possible outcomes, right? And if we look at these examples, like we have different sets. In these cases, our sets are, for instance, A and B. And so we can think about like an event. If we think of an A event occurring, right? If event A is going to occur, it's going to occur. It's somewhere in one of these spaces, right? So if you are in this sort of theoretical space, you'll get an A. If you're in this sort of theoretical space, you'll get a B. And if you happen to be in the middle here, you could get both A and B to occur. And as we look at these sets of outcomes, the A set and the B set of outcomes, understand that the size of each of these A and B circles represents the proportion of the total set that they might make up. So you can see here A is making up a much larger set of possible outcomes than the B set is making up. So that's how we want to think about it. We think about it as sets, right? The A set and the B set. And so they can be described in various ways. There's notation involved here. So if we want to say, B is a subset of A, we would notate it in this fashion. If we're talking about the intersection of two sets, of set A and set B, we can see it's notated here. And then we have the union of both sets. We've got A and B. This includes all the possible places where we could have either A or B, and is denoted here. If we were to talk about A and then the complement, the set complement of A, so everything else except for A, right? That's A with this C notation. And then if we were talking about two sets in which there is no overlap, in which there is no intersection between them, we can call them disjoint, and that is this indicator here, this not. So what are some of so keep in mind, like when I talk about events, and we talk about sort of this, whether or not they're going to co-occur or occur, we're thinking about it in this way. So this is like the picture of how you can sort of like conceptualize all of the A events and all the B events. Like are they gonna, could A and B happen at the same time? Do they always happen at the same time? Are they only going to happen part of the time? Are we only looking at the A and B events and not all of the other S events, right? None of the stuff in the white. Are we just looking at everything except for A? These are important concepts. It's the vocabulary and the conceptual framework we're going to be using for talking about probability. And when we talk about probability, what we're talking about is the proportion that one set of outcomes may make as part of the larger set of outcomes. So what percent of the space is taken up by A as opposed to B, or what percent of the space is taken up by the simultaneous outcomes of A and B, what percent of the space is taken up by the complement of A versus A itself, right? Each of those proportions is actually going to represent, proportion of space is going to represent the actual probability that we're going to calculate. So the more space, then the more number of outcomes we observe within that S space, the higher the probability. So when we think about probability, what is probability? We talked about earlier about the probability of precipitation. A lot of times we think of it as being somewhat equivalent to percentage, right? Except that it's not on a 0 to 100, right? Probability is going to be on a scale of 0 to 1. And if you have a probability of an event occurring equal to 0, that basically means that is completely impossible that event would ever occur. If you have a probability of 1 for an event, that means that event will always happen, right? So that's the scale we're looking at. So generally when we're talking about probabilities, we're usually between 0 and 1, but you can't have a probability that equals 0 and 1. You just have to realize what it represents. Some other basic rules for probability are that the probability of an event can be calculated as the ratio of the number of outcomes, right? And we could call that, say, set A, right? The number of possible outcomes, or the number of outcomes that occur, divided by the total number of possible outcomes, right? So when all outcomes are equally likely. That means that if I wanted to calculate the probability of the complement of A, I can just take that number divided by the total and subtract it from 1, and I can get the complement of that. So that tells you that a set, is always on a scale from 0 to 1. So probability represents some sort of total amount that's equal to 1. And if I want to get everything that's left over, I can just subtract my probability from 1, and I can get the complement of that probability. Another thing to understand is that if I was to think of the union of all the possible outcomes, so if we're talking about it in this case, we're talking about here the union of all possible outcomes, like A and B, right? Then that's going to be the probability A plus the probability of B plus the intersection minus the intersection of A and B. So if you want to think about this case, it's the probability you get all these outcomes, because they overlap, you have to subtract that amount. And then the last thing to understand too is that if A and B are disjoint, then the probability of the union is just A plus B. So if we want to go back here, we can see this is the probability that if they're two disjoint, then I don't have to subtract anything in the middle because they don't overlap, right? So these are just some of the basic rules of probability, but we can leverage these rules later on, and you'll see we're actually able to use these to do some to derive some useful relationships for us later on in how we might calculate probabilities. So there are a couple different types of ways of approaching counts. And remember, like each set, we're talking about the count of the number of outcomes, right? And so sometimes the order matters when we're counting, and sometimes the order doesn't matter. So first, we're going to talk about cases where the order of counting matters, right? And in this case, the reason why we're talking about first is because we are going to be dealing a lot with things like sequences, whether it be amino acid sequences or nucleotide sequences, right? And the order obviously matters in those cases, like the identity and the order of those things matter. And when we think about an ordered selection, what we generally refer to that is we call that a permutation. So that's an ordered selection. Another important idea to understand here is the concept of the multiplication principle. So the multiplication principle basically states that if there are A ways of doing something and B ways of doing another thing, then there are A times B ways of doing both things. So when I think about the multiplication principle, there are ways that I can apply this to questions such as those related to codons, right? And one of the ways I could do this, I could say, okay, well, how many different codons are there, right? And if I was going to do this, basically how I could calculate that is I could figure out that there are, 4 possible outcomes in the first position of the codon, 4 possible outcomes in the second position of the codon, and 4 possible outcomes in the third position of the outcome of the codon. We can think of rolling that 4-sided dice, 3 times. And basically, the number of possible different codons out there is 4 to the third, right? So that's 64 different cases. So that's the total number of possible codons I could come up with. If I was going to start with how many codons there are, that start with T and then with C, I can think about that dice, and if I was going to run it the first time, the only thing I can get is a T, right? So if I want to think of how many different codons are out there and apply this multiplication principle, right, then what it'll say is there's only one possible outcome for that first position, T. Then in that second position, there would be 4 possible outcomes, and in the last position, there'll be 1 possible outcome. So that'd be 1 times 4 times 1. So the answer is 4. So there's 4 possible outcomes that start with T and end with C. What about codons that don't contain G and have no repeated nucleotides? This is a slightly more complicated case. We can see what does the set look like, right? So we have all the cases that start with that don't start with G, right? And they can't have any repeats. So in other words, they have to have an A, a C, and a T. And given that the first base is an A or a C or a T, then the next base, if our first base is an A, the next base has to be either a C or a T, and then the one after that has to be a C in this case or a T in this case, right? And if it starts with a C, it'd have to be an A or a T, and then of course the last base would either have to be an A or a T in this case, right? And so on and so forth. And so if we were to go through and calculate that, right, we could see that what you end up with are 6 possible different outcomes, okay? But if I wanted to actually calculate this using the multiplication principle, basically I could see that I have 3 possible outcomes in the first position. In this position, I'm going to have only 2 possible outcomes because it can only be one of 2 possible things, right? And at this position, I've got only 1 possible outcome. So it's going to be 3 times 2 times 1, which equals 6. So what these counting methods show us is that if we want to count the number of k-selected ordered items, if we allow repetition, it's just going to be n to the k, right? So this is that, 4 possible outcomes. We had 3 positions, so 4 to the third, right? But if we don't allow repetition, as in the previous case, right, well, we're going to end up as a case where the count is going to be this pnk, this lower denotation, and that's going to show as n times n minus 1 times n minus 2, etc., all the way to n minus k plus 1, right? And we can actually summarize that as basically n factorial divided by n minus k factorial. Right, so this is where we don't allow things to repeat. This is that second case of those codons. So we think of this as identifying, the number of permutations that are possible when we don't allow repetition to concern. So all the unique possible outcomes. In addition to the multiplication principle, it's important for us to also recognize the need for identifying how it differs from the addition principle. So the addition principle is important if outcomes can be divided into non-overlapping cases. So if they can be divided into non-overlapping cases, then the total of number of outcomes is the sum of the number of outcomes in this case, not the product of them. So there's a good case if we were to look at, say, the types of possible outcomes we could have for T53 sequences that have 5 nucleotides between 2 motifs. So in this case, what we'd have is we have this R, which could be either A or G. So we have these 2 possible outcomes, 2 possible outcomes, 2 possible outcomes, times 1 possible outcome, times this is either AT or TA. So times 2 possible outcomes, times 1 outcome, times 2 outcomes, times 2 outcomes, times 2 outcomes. So if I actually want to think of how many possible TP53 sequences have 5 nucleotides between the 2 motifs, right? What that means is that I have 1, 2, 3, 4, 5, 6, 7, 2 possible outcome positions. And I have 2 1-position outcomes. So that's 2 times 2 times 2 times 1 times 2 times 1 times 2 times 2 times 2. In other words, this whole section is 2 to the 7th for the left motif. Then I've got 5 possible outcomes that could be any of 5 nucleotides. So that's just going to be 4 to the 5th. And then I've got 2 times 10 to the 7th, 2 times 2 to the 7th power over here as well. So I've got 2 to the 7th times 4 to the 5th times 2 to the 7th. And that's how many possible outcomes that we could have. And it basically equals about 16,770,260. And what this is a classic application of the multiplication principle, right? The same principle we just talked about in the other case. But what do we do if we want to look at all possible combinations? So now we're thinking about non-overlapping cases, right? What about if we have 1 nucleotide in the middle? What if we have 2 nucleotides in the middle? What if we have 3 nucleotides, 13 nucleotides, right? Each one of those cases is a non-overlapping case of possible combinations. So to do that, what we need to do is apply the addition principle. Well, then we're going to have to recalculate and do 2 to the 7th times 4 to the 4th, right? For when there are 4 nucleotides times 2 to the 7th. And we'll have to do 2 to the 7th times 4 to the 3rd, where there are 3 nucleotides times 2 to the 7th. And 2 to the 7th times 4 to the 2nd, right? Times 2 to the 7th, where there are 2 nucleotides. Or 2 to the 7th times 4 to the 1 times 2 to the 7th, where there's only 1 nucleotide. So we have to do this for all nucleotides from 0 to 13, right? And then we add them all up for all the possible calculations. And so while there may be 16,770,216 just for the 5 nucleotide case. Overall, there'd be, let's see, like 1,498,240 possible outcomes for this motif. So that's the total number of possible outcomes for T53 DNA binding motifs. So you can see this is a case where we use the multiplicative principle and we use the added principle to see all possible combinations. So in addition to ordered counting, there are also places where we can use counting methods where the order doesn't matter. And you might ask, well, why is that important? Well, we can use these sort of cases of unordered selections or combinations and manipulate the properties of an equation like this binomial equation shown here in bioinformatic techniques, and allows us the ability to sort of calculate the combinations of ordered sequences such as DNA sequences in some clever ways. And so we'll go over that in class and talk in a little more detail, but just know that we do have this way to identify these unordered sequences. Along with understanding how to calculate the number of outcomes in sets and in parameter space, it's really important that we also understand what to do when outcomes are not equally likely. This happens in real data. And so to do that, we're going to explore some implications of that using sort of our original general equation of the probability of the union of A and B being equal to the probability of A plus the probability of B minus the probability of the intersection of A and B. And just to remind you guys, that intersection means where we have both outcomes being A and B. So this is pretty straightforward to do when it's independent. If the occurrence of one event does not basically depend on the other event, so they're independent of each other, then the probability of the intersection is easily calculated, right? It's just where they intersect, it's just the probability of A times the probability of B. When it becomes dependent, that's where it becomes a lot more complicated. And this is an important case because when we think about it, data in the real world is not always independent. That's kind of a misconception to think that it would be. And so if they are not independent from each other, we call them dependent. And often in that process, we're going to define those types of probabilities using a conditional probability. And so a conditional probability basically quantifies the probability of one event based upon the status of another event. So the probability of A given B, right? And we can use an equation for this relationship, saying that based upon that previous example, the probability of this intersection of A and B, right, equal to the probability of A times the probability of B, which we saw back on this slide, right, down here. We could say, well, if it's a dependent case, right, it would be the probability of A given B times the probability of B equals to that intersection. And if we wanted to get what this conditional probability is, we just sort of manipulate this equation, and now we just have the probability of the intersection of A and B divided by the probability of B. Examples we could use here, including CPG islands in the genome and nucleotide frequencies, as well as the color blindness of sex. So if we use the probability of, say, color blindness in different sexes, where we know that there's bias in the data, so men are much more likely to have color blindness than women, we can end up with this interesting case, right, where we can use this kind of equation of the probability of A given probability given B equals probability of the intersection of A and B divided by the probability of B. So if I just know these numbers, right, it's kind of easy for me to calculate, like, what is the probability of color blindness given that you're a woman, right? Because I know that there are, say, 50,000 men and 50,000 women in this, so I just need to look and see, okay, well, 10 divided by 50,000, right, is going to give me this conditional probability pretty easily. And notice that there's going to be a difference between the two. But I can also calculate, so I can calculate it independently. But say I didn't, have those same numbers, I can also calculate this by using an additional equation, right, where I have, I'm trying to identify the intersection between these two divided by the probability of being a woman, right? So just the probability of being a woman is going to be, 50,000 divided by 100,000, whereas the probability of being colorblind and being a woman is 10 in 100,000. And so if I just take those two and I divide them from each other, basically I end up with 10 divided by 50,000, which is exactly the same thing I would have gotten if I'd done this process over here on the right side. So the reason why I bring up these ideas of conditional probabilities is, one, they're real, they happen all the time, and we should be able to compensate for them. Things are not always independent in real life. But the other reason why is that it opens us this sort of door for us to talk about Bayesian probability and statistics. Specifically, we're going to be looking at Bayes' law. And Bayes' law is basically just a rule that we can use to compute a conditional probability. So in the last case, right, we were interested in looking at the probability of A given B. Right? In this case, we're going to be more focused on the probability of B given A. And that's really at the heart of Bayesian inference. So, we use Bayesian statistics already all the time. We use them in, interpreting positive tests for disease. We use them in bioinformatics all over the place, especially in things like multiple sequence alignments, using hidden Markov models and things of that sort. Phylogenetics, it's very commonly used approach, but it's not always a commonly taught method. So we're going to spend some time and just talk about it so you guys can have a good general sense of how to approach it and understand it. So, the basics of the Bayesian perspective lies in Bayes' formula, which basically says, what is the prior probability, what is the probability of hypothesis given the data, right? So that's what we're trying to figure out. And basically the equation says, well, if you're trying to figure out what's the probability of the hypothesis given the data, then that's going to be equal to the probability of the data given the hypothesis times the probability of the hypothesis, which we often refer to as the prior probability, whereas the probability of the hypothesis given the data is referred to the posterior probability. That probability of the data given the hypothesis, we refer to that as the likelihood. And then the last thing that we're going to be looking at is what's the probability of the data. Right? So we take the product of the probability of the data given the hypothesis times the probability of the hypothesis and divide it by the probability of data, or the marginal likelihood of the model, and that takes into consideration sort of like the total amount of possible probability in a sample. So the common example right here is what's the probability of getting, coin flips. We love coin flips and probability examples. So what's the probability of getting ahead in a coin flip? There's kind of two ways to think about it. There's the traditional way, and we'll say, well, yeah, it's 50%, right? Like that's what it's going to be. That's what the probability is of getting ahead if it's a fair coin. Well, that's what frequentists believe. So people who aren't Bayesians, who believe in traditional statistics, they have this idea that, there's this long-term frequency. Like there's this value that's associated with flipping coins, and it's 0.5 if everything's fair, right? Like there's some sort of rule to the universe that 0.5 will always be the value. But, probability can be a lot more subjective than that. It doesn't always have this long-term frequency. It can be contextual. It can be all kinds of things. And so, we can use previous information to drive our understanding of what's feasible. We can use other knowledge about theoretical considerations or context and environment. There's all different kinds of ways that we might say, well, this stuff actually matters in how we're going to calculate our probabilities. And so what it says is that, context and previous knowledge are actually really important. And if we think about how we make decisions as humans, we apply Bayesian concepts to so many things in our lives, right? We look at what was successful in the past, what we know about systems, and we use that to guide our decision making. And so a big part of the Bayesian approach is understanding that you are going to be using prior knowledge. So again, this probability of the hypothesis, which is identified as the prior probability. What it does is it puts us some boundaries on the amount of, on the set size that we're looking at. It puts some expectations on what we expect to get out of different types of analyses. And so it's really incredibly useful, and it can, you can know different levels of things prior to going to experiment. You may know a lot or you may know a little, but using Bayes, you can take that and make it into something really useful in your data analysis. And a lot of people think that that Bayesian probabilities actually make a lot more sense, and that this is like how the true world works, and it's expressed in degrees of belief or probabilities. And so, these two perspectives are always have often been at odds in the past. I think, I'll just tell you right now, like as a scientist, I find merits in both of them, and I use both of them. There are strict frequentists and strict Bayesians, and to each their own. When you think about, what is the frequentist inference approach, right? It never uses or gives the probability of a hypothesis. There's no prior or posterior probability. It depends on the likelihood, so the probability of the data given the hypothesis for both observed and unobserved data. So it's actually making a claim about things that you haven't observed, which some might say is more powerful and some might say is more biased, right? It doesn't require a prior you don't have to have any previous information about your system before you take a frequentist approach. It's been the dominant statistical practice for a while now, and it's not as computationally intensive as Bayesian approaches. Whereas with Bayesian approaches, what you're actually doing is you're using probabilities for both the hypothesis and the data, right? So there in the frequentist approach, there's no probability associated with the hypothesis, right? In this case, there is. Like, what is the probability of the hypothesis? It really depends, on these really important components called the prior, right, which is your knowledge about a system going into it, and the likelihood of that observed function. So that same probability of the data given the hypothesis. It requires one to know how to construct a good subjective prior. It dominated statistical practice actually back before the 20th century. This is not a new thing. It was actually an 18th century Reverend Thomas Bayes who was the first one to propose this probabilistic framework. But it fell out of favor after a certain amount of time. Due to the fact that like Bayesian approaches often involve lots of parameters, it can be kind of computationally intensive to do. So some of the critiques of frequentist inference is basically that you have to have experiments that have to be fully specified ahead of time, or else they don't really fit a frequentist framework. And so there's lots of opportunities that are lost in sciences because of that. And it depends on the exact experimental it depends on an exact experimental setup and some ad hoc assessment of that. For Bayesian, the biggest complaint people generally have about it is the role of the prior, and that how we pick priors is kind of a non-standard process, and it can have a huge influence on what the outcome is of your analysis. So you got to be careful. Also, there's this problem with the hypothesis traditionally being viewed as being either true or false, regardless of whether one knows which it is in that case, right? But in Bayesian approaches, we're often looking at support for different hypotheses. So it's a different way. We may not be completely rejecting some of these hypotheses. So for those of you who love the frequentist perspective, basically people get behind it thinking, well, it's objective, right? So we're not using prior information, we're not biasing in that fashion. That it explicitly describes the trade-offs between, false negatives and false positives with regard to a null hypothesis, right? So it's got a very clear framework and well-accepted framework. And that it basically involves a lot of care that's specifically targeted towards dealing with experimental bias. In terms of Bayesians say, well, the probability of the hypothesis is exactly what we need to make a decision, right? So this is our hypothesis about the system. What's the probability it's right? Like, that's the best bit of information that we could hope to use and when we need to make a decision. And the other nice thing about it is that there's no requirement for every contingency to be planned for ahead of time, right? Like, Bayesians can reuse their information in a future analysis, right? So we can retack and reuse it so your current posterior distribution then becomes your future prior distribution. And so it can be guided in that fashion. When thinking about Bayes' law, it's important to understand how we're actually going to compute our conditional probabilities. And to do that, we have to take into consideration the law of total probability. In this case, total probability is going to be defined of a specific event. It's going to be computed through the calculation of a weighted average of conditional probabilities. So in order for us to get the probability of A and consider this entire set space appropriately, we really do have to, weight what is the probability of A given each of these different B possible outcomes, right? They each make up different space and they have different dependent relationships with A. And then, if we're going to look to further apply Bayes' law, one of the main outcomes, right, is this idea of understanding the probability of B, or your hypothesis, given the data, right? That's what we're one of the questions that we're trying to determine, right? What is that conditional probability? But part of that is we kind of have to know what the probability of A is given B, and that can sometimes be complicated. And so, in order to understand these cases, a lot of times what we're actually doing, if you want to think about why we need to know these total probabilities, because if I'm going to look at the outcome of, say, what is the probability of, say, the B3 hypothesis given the data, I need to only look at possible outcomes for A, right? Like, I only need to look for the outcomes for A and then see, well, what proportion does the intersection of A and B3 account for? And that's going to help tell me how much support there is for B3, because if B3 accounts for like a huge amount of the proportion of all the possible A outcomes, then that's going to help me understand that this hypothesis is much more likely than other hypotheses. And so if we think about it, what we're actually looking for is, if we look at the probability of B sub I given A, we're actually looking at what is the probability of the intersection of A and B, in this case, divided by the total probability of A. And if I was to break this down further, what that ends up being is, if I'm going to look at the probability of A given a specific BI, so this could be B3, B2, B4, whatever, right, times the probability of BI, right? And why does this equal this? Well, that's because if we go back, remember we had our equation all the way back at the beginning, which said the probability of A given B times the probability of B equals the intersection between A and B, right? Right, remember we had this where it's the probability of the intersection of A and B is probability of B times the probability of A given B. And then if I'm going to say what is the probability of A in this case, right, the probability of A is going to be equal to the sum of all the probabilities of the intersections of A and B, so all of these, right? So all the possible A events where B and A occur. And to calculate each of them, right, I'm going to take this equation up here and I'm going to add up all of them together. And that gets back to this equation, right? Like we're then applying this equation. So we substitute in this value into this position on this equation. So it all comes back together to end up with this equation, which ends up being pretty similar to what we had here before, right? When we talked about Bayes' formula, the probability of hypothesis given the data. And then we have our likelihood, our prior, and our total marginal likelihood, right? Our total probability on the bottom. And that's what we're looking at here, right? We've got our probability of hypothesis given the data. We've got our likelihood, our prior, right here, our prior probability of this hypothesis. And then on the bottom here, we have our total probability or our marginal likelihood. And note that what we're doing here now is calculating the proportion of the intersection of A and B and how much that makes up of the total probability for the probability of A, and that will give us the support of the data for this one hypothesis. And so that's the idea of the Bayesian perspective, and it's very unique, right? Because we're looking at the support for different hypotheses, so I can evaluate whether there's more support for the B hypothesis or the B3 hypothesis or the B2 hypothesis or the B4 hypothesis. There's lots of different opportunities for me to explore how well the data supports different hypotheses and to put it into a rigorous statistical framework, right? And this doesn't necessarily get to me rejecting any one hypothesis, like the frequentists would argue, which might be detrimental in the long run because I can't just say I can't get rid of hypotheses in some way. But this type of reasoning, which is very abductive in its approach, feels kind of satisfying when we think about what we're dealing with in terms of our ability to actually do true frequentist work and all the ways that modern science and complicated and complex systems can violate or make it really difficult to accomplish that work, but still provides us a rigorous framework for evaluation. There's just some simple things here. Like if we take a simple thought question, like, we've got some cells were staining in the lab, and we've got, new cells staining at a certain probability, high probability, 95%, 1-day-old cells at 90%, 2-day-old cells at 80%. We have a mix of those sort of cells, like 60% of new cells, 25 of old, 15 of 2-day-old. We can calculate what the probability of the selected stain will stain properly, but just using a Bayesian perspective, we can say, right? We can say, well, I can probably figure out the range that I would expect cells to properly if I pick a cell at random, like what range it must lie in, right? That range, I only have new and 2-day-old cells, so that range is probably going to be between 0.95 and 0.8, right? Like that's the probability range that I'm thinking of. Why not use that information? Like if I already know that, and I already understand that, and I believe that data, why not use that to help guide my experiment and not have me waste time exploring parameter space that doesn't matter to the question that I'm interested in? So there's some logic in there, right? There's some truth in there. Now, it's not always that simple, but it is, I think, an important point for us to consider and a powerful tool for us to use in our analyses. So we'll be practicing and going over more of these examples in class so you guys can get a better sense of how the numbers actually plug in here. But I just wanted to give you some background explanation for before we actually do practice together. All right.